Next morphology portion, the wings. The insects are the only invertebrates which are characterized by possessing the wings, except Pterygota and some secondarily wingless pterygote. All modern insects wear laterally a pair of wings on each naso and metathorax and are commonly called as the fore and hind wings, respectively. Both the pairs of wings may perform the flight as seen in a majority of insects, or one pair of wings may modify into altogether different structures like the halters found in Diptera and Strepsiptera and Electra or Tegmina in Orthoptera, Diptera and Coleoptera. Origin of wings. The wings are of great phylogenetic and taxonomic importance. The origin and evolution of wings represent, in true sense, the evolutionary history of insects. On the basis of fossil records, wing venation and development of the wing skeleton, musculature, and wing buds of various insects, large number of workers have tried to trace the origin of wings. The most popular theories are given below. The paranotal theory. This theory is also coined as the flying squirrel theory, as it was first put forth by Miller in 1873 to 1875. According to the paranotal theory, also called the flying squirrel theory, the wings are the dorsal backward prolongations of the pteronota. They are evolved from the rigid, double-walled lobe-like lateral expansions of the thoracic pteryga and paranota or paranotal lobes. The fossil evidences reveal that the lateral lamellae on the Paleodictyopteran abdomen, marginal subcoxy, as well as the prothoracic Paleodictyopteran wingless typical paranota, are homologous with the wing pads of the modern insects, and all the structures show venation alike that of the wings, supporting strongly the paranotal theory. The paranotal theory is recently supported by Sharrow, Hamilton, and Wharton, and some other scientists. It is well evident that some of the Carboniferous insects were bearing the paranotal lobes on the prothorax in addition to the fore and hind wings, suggesting that the wings primarily evolved as a paired thoracic organ, while those of the prothorax were lost later on. Then next theory, the pre-truncation theory. Some other scientists will develop this theory after establishing homology in wing venation among various orders of insects. The main principle of the pre-truncation theory is that the truncation in the nymphal wing pads recapitulates the wing venation of their adult ancestors. They have further suggested that the truncation alone determines the position of the veins. So other scientists found that veins in the nymphal wing pads are formed from the lacunae in which both the trachea and nerves grow only after their pattern has been established. It is now well established that the blood channels play a primary role in determining the venation rather than the truncation was as revealed from the microanatomical, teratological, ontogenetic, and experimental studies. The veins are secreted by the epidermal cells through deposition of cuticular material above and below the lacunae only at the final adult molt and are in no case formed or derived from the trachea. Secondly, veins may fuse and migrate, but the trachea never undergoes such modifications in the wings. Then the spiracular flap theory. Some other scientists have proposed the spiracular flap theory on the consideration that wings are originated from the integumental imaginations, which literally serve as the spiracular flaps. From the wing development of Odonata, she has suggested that the small spiracular flaps have achieved first mobility by acquiring the molecular and sensory mechanisms to perform respiratory and tactile functions. Later on, the large folds have been developed in order to protect the spiracles and form a chamber around each spiracle. With the time, they have been enlarged and become mobile, performing the function of protective coverings. Lastly, they become large enough to serve a ventilating mechanism for the tracheal system and in addition started to help the forward locomotory movement. The evolutionary process finally has modified these structures. in the locomotory organs by providing powerful musculature and large surface area with a high frequency of beating. So this theory is such as much debatable in relation to the distribution of spiracles in Pterygoda. There is no resemblance in the total number of spiracles in the modern and fossil insects. No modern insect shows ontogenetic recapitulation of the molecular spiracular flap-like structures into wings. And the stylus theory has proposed a novel hypothesis that the wings might have been originated from the coxal stylia of the Pterygoda. He has suggested that homology in between the abdominal and coxal thoracic styli of pterygotes with the abdominal gill plates or mayfly nymphs and wings of the pterygotes. Moreover, development pattern of the gill plates and that of thoracic wings is very much alike, and both of the structures exhibit true venation, articulation between subcoxa and tergum, and their innervation by the subcoxal muscles. The homology in between gill plates and wings has been strongly supported. Then the fin theory. Fin theory of wing origin. According to him, Bradley, according to him, the pre-flight wings were too small to support the insect in air, and later they have been evolved into the true wings by acquiring essential characters such as increase in size, development of venation, flight musculature, and basal articulation. The ancient terrestrial pterygotes have become an amphibiotic, adapted for excursions in the water. They were, however, capable of leaving the water for the purpose of mating and dispersal. In his opinion, the pre-flight wings or prow wings could have been highly useful as the propulsion organs or the fins in the water. In the process of using prow wings as the fins, the hinge and flight musculature might have started to develop. As the aquatic nymphs of Ephemeroptera and Odonata have closed type of respiratory system, the spiracles are mechanically closed and fin-like structures have been developed into the wings with switching over their environment. And the gill cover theory. The gill cover theory has been proposed long back in 1906 by Woodworth. It bridges the spiracular flap theory and the fin theory. According to this theory, the origin of wings is assumed to be conditioned by change of habitat from terrestrial to aquatic to aerial. Some of the scientists consider the abdominal gills as the gill covers found in some mayfly nymphs, which actually function as the opercula for the filamentous gill clusters. These covers are stiffed by a real venation, and the veins are even provided with rows of spines as in the flying wings, and in the final molt these covers develop into the wings. Then morphological variations. A pair of long hind wings in Diptera and that of four wings in Strepsiptera are reduced greatly and modified into the dumbbell-shaped structures, the halters. In some groups of insects, both pairs of wings are lost completely, providing upstairs condition and secondary adaptation to achieve parasitic mode of life, as evidence in Socoptera, Malophaga, Anoplura, Siphonaptera, and some Hemiptera. Both pairs of wings resemble one another in their size and shape in only few groups of insects, such as Zygoptera, Isoptera, Mecoptera, Embioptera, while in other insects, the wings of one pair differs from that of the other pair in morphology. The hind wings are generally larger than foreones as they are provided with the large venal lobes. Next, the wing structure. During post-embryonic development, the wings develop as the external lateral wing pads on either side of the meso and metathorax in the exopterygotes, and they develop in the form of wing buds internally in the respective thoracic segments in the endopterygotes. These structures are evident as the integumental lateral extensions of the combined pleura and nota of the pterothoracic segments. In the adult insects, the wings appear as thin, opaque, transparent or leathery, partially pigmented or darkly sclerotized fan-like, flattened, membranous structures. The wings bear a group of sclerites at their base, a complex of longitudinal and cross veins throughout the wing body, and various types of sense organs and pigments. The basal sclerites perform articulation of the wings and with the thorax as they rotate with one another, and also innervated by the thoracic flight muscle. The wings bear a specific pattern of venation which is derived from unique arrangement of veins. The veins provide mechanical support and folding to the wings. The sense organs are mostly concentrated at the base of the wings and proximal part of the wings. They play important role in determination of direction of the wing movements and navigating through during flight. The wings have often smooth dorsal surface, but in Lepidoptera they are covered with the scales, while in Trichoptera and Diptera with the hairs which provide the brilliant pigmentation and hairy covering respectively. The wings are somewhat triangular in shape and possess three distinct margins: the anterior costal margin, the outer or apical margin, and the inner or anal margin. The costal margin contains a pigmented spot, the pterostigma or stigma in large number of insects. The wing base. In Ephemeroptera and Odonata, the chitinous wing base is continued with the lateral wall of the thorax, while in rest of the insects, several sclerites are uniformly grouped at the wing base of each wing. They form a composite joint by which the wing articulates with the thorax. It includes the tegula, axillary cord, axillary sclerites, and variable forms of skeletal plates. The tegula are mostly confined to the forewings, particularly in Lepidoptera, Hymenoptera, and Diptera. Each lies at the base of the costal vein as a small hairy chitinous pad or a scale-like sclerite. It may occur in some insects in the form of chitinous plate overlapping the wing base. And the axillary cord. It is a lobe-like structure lying closely with the vernal margin in the posterior region. It often appears as a ligament and known as the articular membrane. Then we have axillatories. First, axillary lies at the base of the subcostal vein. It articulates with the anterior notal wing process and sometimes divided into two parts. The second axillary accommodates at the base of the radius vein. It articulates with the first axillary proximally and with the wing process of the pleuron distally. The third axillary lies between the base of the anal veins and the fourth axillary. It articulates directly with the posterior notal wing process. The fourth axillary is present only in Orthoptera and Hymenoptera. It articulates with the posterior notal wing process proximally and with the third sclerite distally. And the median plates, they occur in pair. One of the median plates is associated with the bases of the media cubitus and the first anal veins, while the other is often fused or closely opposed with the third axillary. Both the median plates are separated by the oblique membranous fold. And the humeral and axillary plates. In Odonata, besides the anterior well-developed humeral plate, there is a posterior plate, the axillary plate, at the base of the four postcostal veins. It articulates with the posterior half of the lateral membranous margin and with one of the arms of the pleural wing process. Both the plates, humeral and axillary, are slightly articulating with one another. All skeletal components described above are embedded in the double-walled membrane. The basal wing membrane is fused with the pleura by a resilient, rubber-like ligament, or also called as the wing hinge. The wing areas or regions. The entire body of wing is differentiated broadly into the basal articular or axillary region and rest of the alar region. The alar region can be divided into three subregions which are distinctly separated from the folding lines in several insects. The vernal fold separates the anterior region, remigium, from the posterior region, and vanus. Similarly, the jugal fold differentiates the jugum, lobe-like area, from the vanus. The axillary region is a triangular in shape and includes the axillary sclerites mostly related to the articulation and receives the neuromuscular, respiratory, and circulatory accessories of the thorax. The remigium is provided with the costal, subcostal, radial, median, cubital, and postcubital veins and represents the major area of the expanded wing. The vanus is represented by the area occupied by the vanal veins, which was often well developed in the hind wing. In Diptera, a pair of membranous lobes at posterior margin of wing base are predominantly evident and are known as the outer and inner squamata. They constitute the area known as alula. It is well developed in the housefly. Then wing venation, when you talk about that, the veins are also known as the nervous and represent the rib-like structures of the wings. These are hollow, tubular, sclerotized structures and each is typically provided with a nerve, trachea and a circulating fluid. The precursors of the veins in the nymphal wing pads are the lacunae, free spaces which are surrounded by the spongy columnar epithelial cells. According to Holdsworth in 1940, trachea and nerves grow into the channels only after their pattern has been established. The veins are secreted by the epidermal cells through a deposition of cuticular material above and below lacunae only at the final adult molt. The wing venation represents the complex organization of veins or nerves within the wing. The wings may be unbranched or branched, isolated or jointed with one another by cross veins and convex or concave on the basis of morphological characteristic features. The representing simple system of venation, the K-type venation, and it can be described as follows. The costa. It is the first anterior marginal vein due to which the anterior margin of the wing is commonly designated as costal margin. It starts from the humeral plate at the wing base and runs towards the outer origin of the wing without giving branches during its course. That is the costa. Without giving branches during its course, it is the convex vein and may bear sometimes the stigma as can be seen in the fore and hind wings of Odonata and only on the forewings of Hymenoptera, Socoptera and Mycoptera. The subcosta. It is the second vein and distally divided into two branches. The outer and inner branches are usually designated as the SC1 and SC2 respectively. It is the concave vein. It starts from the distal end of the first axillary sclerite at the wing base. At the site of branching, it forms a sort of fork as both the sclerites are widened far away from one another. In the absence of costa, it represents the first vein of the wing in some insects. The radius, it starts from the anterior end of the secondary axillary sclerite at the base of the wing and represents the third major vein of the wing. After running for a short distance as a strongly sclerotized convex vein, it divides first into two branches. The outer branch runs directly towards the outer margin. The second branch is termed as the radial sector, which appears to be the concave and gives origin to four branches, the R2, R1, R4 and R5. The media, it is the fourth major vein, starts from the distal medial plate and mostly fuses with the radius. At the base, it largely sclerotized and convex, and after running for a short distance, it gives two branches, the anterior and posterior media, respectively. The anterior media divides further into two convex branches, the M1A and MA2. The posterior media, on the contrary, is the concave and sometimes also known as media sector. It provides terminally into four branches, MP1, MP2, MP3, MP4, either at a time or discontinuously. In Odonata, M4 is present, while in other higher insects only the MP is existing, falsely representing the media as a four-branched vein. The cubitus, it is the fifth vein and starts from the distal medial plate at the wing base. At the beginning, it seems to be a convex vein, but later on it divides into the two cubitus and the second cubitus, Q2, which are convex and concave. The first cubitus may ramify into the terminal branches, the anterior first cubitus and posterior first cubitus. The anal veins, these veins start from a close association of the third axillary sclerite of the wing base and constitute a group of veins in the anal region of the wing. The anal veins are also termed as venal veins. The anteriormost anal vein, which lies just behind the cubitus, is termed as the postcubitus by some workers. The number of anal veins varies from 1 to 12 among different groups of insects. These veins are generally convex and unbranched, but some of them may divide into sub-branches in rare cases. Then the jugal veins. These veins occur in the jugal lobe of the wing. Their number may differ invariably, but commonly are two, the first and second jugal veins. These are short, unbranched and concave in their appearance. The cross veins, the various types of longitudinal veins described above, are linked with one another by the cross veins. The cross veins run in vertical plane and are confined to the adjacent longitudinal lines only. From their location, these veins are termed as humeral cross vein lying between the costa and subcosta. Radial cross vein lying between the first and second branches of radius. Sectorial cross veins lying between the sub-branches of second or radius sector. Then radiomedial cross veins lying between the radius and media veins. And media cross veins lying between the branches of the posterior media. And media cubital cross veins lying between the media and cubitus. Then there are some portions called archidiction in Paleodictuta. The longitudinal veins are netted by distinct cross veins, forming an irregular network of veins called archidiction. So, according to Comstock and Needham and Bradley, the archetype venation described above undergoes a lot of variations among different groups of modern insects due to the reduction or addition in the number of veins. The veins may degenerate, atrophy, or fuse together, and therefore it becomes very difficult to make homology in between the wing venation of even closely related groups of insects. Similarly, the presence of large number of veins in some insects occur mostly due to either multiplication, duplication, or enormous branching of the principal veins. Due to unique distribution of longitudinal and cross veins, the wing area is divided into two large number of small spaces or cells. This can be divided into two types: basal cells lying towards the base of wings and the distal cells lying in between the branches of principal veins. The venation of wings is greatly modified in different groups of insects. Then wing trachation. Each instar possesses its own tracheal system which may more or less differ from that of the next instar and from the adult stage. The tracheal pattern is not necessarily homologous between instars because new additional tracheal branches are formed and the old ones are discarded. According to some scientists, the generic or family level wing trachation in some insects is stable enough and helpful in the study of venation pattern. It should be noted that the particular tracheae which serve the wing never migrate or fuse to the veins can migrate and has been erroneously brought forward by some other scientists. In almost all insects, the fore and hind wings are served by two tracheae. The anterior trachea, which supplies oxygen to the costomedian venation, and the posterior trachea, which supplies the cubitovenal venation. The exception is the only Ephemeroptera in which all veins include the cubitoanal system and served only by the anterior trachea from the posterior spiracle. This condition is whether primitive or secondarily derived is still questionable. Then wing coupling apparatus. In several insects, coupling of the fore and hind wings of either side takes place in such a way that they move together as a single unit, mostly with the help of lobes or spines lying in the wing base. The jugal lobe at the base of forewing and the humeral lobe at the base of costar margin of hind wing form a simple or primitive type of apparatus in some Mecoptera. Both lobes contain setae, the humeral lobe and jugal lobe. Okay, so humeral lobe specially bears a frenular bristle. From this primitive type of wing coupling mechanism, complex types, jugate and frenate types have been evolved in some Neuroptera, Trichoptera and Lepidoptera with the development of long jugal lobe on the forewing or that of frenulum and retinaculum like distils. So among Hymenoptera, hamulate type of wing coupling consists of a row of small hooks on middle costar margin of the hind wing and the cuticular fold along hind margin of the forewing is developed. In some Lepidoptera, amplexiform coupling occurs as found in Papilionaria, in which the wings are coupled by simply overlapping basal. Primitive jugate frenate in case of Lepidoptera and hamulate type. Hamulate is seen in Hymenoptera.